Hello my dear children, welcome back to BYJU'S. My name is Sneha Sharma and we are in the process of discussing NEET 2020 paper. I hope you all are ready with your books and pen. I hope you have been practicing the questions. So this is to understand how exactly are we going to attempt the questions when it comes for final exam. तो चलिए start करते हैं अपना ये session. Let us have a look at this question. Which of the following is correct about viroids? ठीक है, तो यहाँ पर हम बात कर रहे हैं viroids की. You know that the smallest known infectious particles as such when you talk about are viruses. लेकिन कुछ ऐसे particles होते हैं जो viruses से भी छोटे होते हैं. अच्छा, viruses में और viroids में exactly what is the point of difference? Viroids जो होते हैं, these are like literally speaking, they are naked RNA particles, right? They are naked RNA particles which are known to cause infection in plants. So they literally become plant pathogens. है ना? Viruses का में फर्क क्या होता है? Viruses में genetic material जो होता है, it can be either DNA और RNA. And ये जो DNA और RNA होते हैं, इनके surrounding में there is a presence of a protein coat, right? So let me show you an image because of which I think it will be very clear to you all. तो ये जो है, this is a representative image of a virus. Okay, virus as I said is one of the known smallest infectious particle. Of course उससे भी छोटे size का होता है viroid. And ये जो difference है ये क्यों आ रहा है? क्योंकि viruses जो होते हैं इनका genetic material या तो DNA या फिर RNA होता है. मतलब it is not going to be both of them, but it is going to be either a DNA virus or an RNA or a basically ribonucleic acid जिसमें genetic material होता है वो वाला virus होता है. And इनके surrounding में there is usually going to be something called as a capsid, which is usually a protein coat, right? So this is the envelope that I am talking about. This is what is the capsid. इसके ऊपर कुछ रिसेप्टिव ग्लाइकोप्रोटीन्स भी लगे हो सकते हैं या कोई और स्पेसिफिक टाइप के रिसेप्टर्स लगे होते हैं. सो दैट बिकम्स अ कंप्लीट वायरस. अब वायरॉइड क्या होता है? वायरॉइड जो होते हैं वो नेकेड आरएनए पार्टिकल्स होते हैं. इनके सराउंडिंग में कोई भी प्रोटीन कोट वगैरह नहीं होती है एंड यूजुअली ये प्लांट पैथोजेंस होते हैं. वन ऑफ द बेस्ट एग्जांपल्स दैट यू हैव लर्नड अबाउट इज समथिंग कॉल्ड एज PSTV, राइट? दैट इज पोटैटो स्पिंडल ट्यूबर वायरस, वायरॉइड, या फिर इससे जो डिजीज कॉज होती है उसे हम बुलाते हैं पोटैटो स्पिंडल ट्यूबर डिजीज, PSTD. All right, do not get confused with this term which is actually used in one of the human medical terms as well, but PSTV यहाँ पर stand करता है potato spindle tuber viroid के लिए and PSTD is the condition that it is causing. यहाँ पर होता क्या है कि potato plant, Solanaceae family के जो plants होते हैं, in fact इसमें हम पढ़ते हमेशा potato है but actually sometimes it also infects the tomato plants. तो यहाँ पर क्या होता है, इस पूरे plant की, potato plant की, and hence the size of potato that is formed from these plants which are infected with PSTV is very very stunted. तो इसकी जो ग्रोथ है वो बहुत कम हो जाती है. इसके जो साइज ऑफ पोटेटोज बनते हैं वो लिटरली ऐसे इलोंगेटेड, जैसे थोड़े थोड़े पतले से, छोटे से, उस तरीके के पोटेटोज प्रोड्यूस करते हैं ये वाले इन्फेक्टेड प्लांट्स. सो दिस इज द बेस्ट एग्जांपल फॉर यू टू अंडरस्टैंड व्हाट एग्जैक्टली इज अ वायरॉइड. ऑलराइट, एक बार ऑप्शंस की तरफ देखते हैं. दे हैव DNA विद प्रोटीन कोट. तो DNA विद प्रोटीन कोट अगर किसी हम लोग वायरस जैसे इन्फेक्शियस पार्टिकल के लिए बोलते हैं तो दिस इज गोइंग टू बी प्रेजेंट इन वायरस एंड नॉट वायरॉइड. तो ये ऑप्शन ऑब्वियसली इस क्वेश्चन के लिए सही नहीं है. फिर आता है दे हैव DNA विदाउट प्रोटीन कोड. देयर इज नथिंग लाइक दिस कॉन्सेप्ट दैट एग्जिस्ट विद ईदर वायरसेस और वायरॉइड्स, राइट? देन वी हैव RNA विद प्रोटीन कोड. नहीं, RNA विद प्रोटीन कोड तो हमारे वायरसेस ही हो जाएंगे. सो दीज टू ऑप्शंस बिकम virus for us. This is a non-existing kind of entity. And फिर last option आता है they have free RNA without protein code. So obviously option D for us for this question is going to be the right answer. Let us have a look at this question. The infectious stage of Plasmodium that enters the human body is which of the following? अब ये question काफी ज़्यादा straight forward है and it's a very important question from the chapter Human Health and Diseases. You have to know and understand the malarial parasite cycle, right? This is a very important cycle. बहुत बार इस cycle से question पूछे जाते हैं. So this becomes very 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 important. अब यहाँ पे question की बात रही. So here the right answer is going to be your sporozoites. Okay. So rather than explaining you just this point, let me give you a very quick revision of the whole cycle, reproductive cycle of this Plasmodium, which is a malarial parasite, which is what is carried by a female Anopheles mosquito. So I hope before that you already know that malaria जो होता है वो काफी ज्यादा tropical area में prevalent है because of वहाँ पर इतने ज्यादा because of the presence of so many mosquitoes in those particular areas, right? And when we talk about this whole cycle of Plasmodium, basically it is digenetic, which means what? This term you must have learnt in your syllabus. Digenetic का मतलब होता है जब दो host होते हैं, right? So let me write it down for you here so that if in case you don't know this, you can take this down. Digenetic means having two hosts. There is first host, which is like the primary host, okay? So the primary host for Plasmodium is going to be female Anopheles mosquito, okay? And mosquito, right? Yeah. And the second one, which is called as the intermediate host, ये होते हैं हम humans, right? And that is when it causes malaria in us, right? So intermediate host is going to be humans. जो sexual cycle होती है of the Plasmodium, it is majorly going to be covered in the primary host. So let me write it down here, क्योंकि ये थ्योरी बहुत इंपॉर्टेंट है. और जो इंटरमीडिएट होस्ट होती है यहाँ पर मेजरली एसेक्सुअल साइकिल होती है इस प्लाज्मोडियम की. प्लाज्मोडियम जीनस के काफी सारे स्पीशीज हैं विच कॉजेस मलेरिया. देयर इज फाल्सिपेरम, देयर इज ओवेल, देयर इज मलेरी, देयर इज गोइंग टू बी वाइवैक्स, राइट? फाल्सिपेरम बीइंग वन ऑफ द मोस्ट डेंजरस वंस. तो अब एक बार स्टार्ट करते हैं. मैं आपको बहुत ही सुंदर सी साइकिल दिखाती हूँ. ये इट्स अ ब्यूटिफुल साइकिल विच इज रिप्रेजेंटिंग हाउ एग्जैक्टली मलेरियल पैरासाइट कंप्लीट्स इट्स लाइफ साइकिल इन दीज टू होस्ट एज सच, ओके? तो स्टार्ट करते हैं सबसे पहले तो फीमेल एनोफिलिस मस्कीटो से. तो आपको पता है कि मलेरिया हमें ट्रांसफर होने के लिए सबसे जरूरी होता है हैविंग अ मस्कीटो बाइट अस. व्हिच मस्कीटो? हैविंग अ फीमेल एनोफिलिस मस्कीटो. लेट मी चेंज द कलर हियर. या, this is a female Anopheles mosquito, okay? Now this mosquito is going to just listen to it like a story, okay? आप इसे कहानी की तरह सुनिए. आपको कभी भी ऐसा नहीं होगा कि आप ये भूल जाएंगे. मेरे वर्ड्स को फॉलो कीजिए, जिस फ्लो में मैं समझा रही हूँ उसे फॉलो कीजिए, शायद आप ये कभी इसके बाद भूलेंगे नहीं, ओके? तो एक फीमेल मस्कीटो जो होती है एनोफिलिस मस्कीटो, वो हम लोग को क्यों विजिट करती है? टू कट अस, बिकॉज ऑब्वियसली दैट मस्कीटो इज बेसिकली हंग्री एंड दे वांट टू सक ब्लड, राइट? तो अब क्या होगा, जैसे ही वो हमें दे बेसिकली पियर्स व्हाटएवर kind of appendages it has to start sucking the blood. What happens here is, in order to prevent the clotting of our blood so that the female Anopheles mosquito can literally keep sucking up the blood as much as possible, उसके लिए वो लोग क्या करते हैं? वो लोग अपना सलाइवा हम में थोड़ा सा छोड़ देते हैं. And when this saliva is basically left in us, this contains something called as sporozoids. ओके मैं पूरी लाइफ साइकिल के एंड में आकर के आपको बताऊंगी कि ये स्पोरोजॉइड कैसे बने हैं. आप एंड तक खुद कनेक्ट कर पाएंगे. सो ये जो स्पोरोजॉइड हैं ये फीमेल एनोफिलिस मस्कीटो के सलाइवा में ऑलरेडी प्रेजेंट है. दीज आर स्पोरोजॉइड्स ऑफ द प्लाज्मोडियम, ऑफ कोर्स, राइट? प्लीज रिमेंबर, प्लाज्मोडियम इज अ parasite. It's a malarial parasite which is being carried in the female Anopheles mosquito. So this mosquito is like a vector for us, okay? So coming to this point, so as soon as this happens, what happens next is, यहाँ पे जो स्पोरोजॉइड रिलीज हुए हैं वो धीरे धीरे जाकर के अपने ब्लड में ट्रैवल करके लिवर स्टेज पे आ जाते हैं. मतलब क्या होता है कि दे एंटर द लिवर, right? And once they enter the liver, they leave the bloodstream and enter or invade the hepatocytes. लिवर सेल्स में अब वो इन्फेक्शन कॉज कर देते हैं, ओके? जैसे ही वो लिवर स्टेज पे आते हैं, मतलब ये जो इन्फेक्शन यहाँ पर होता है, यहाँ पर मलेरियल पैरासाइट यूजुअली टेक्स अबाउट लाइक गुड 5 to 10 days in order to continuously allow a lot of nuclear division for it to happen. एंड जब ये बहुत ज्यादा कंटिन्यूसली न्यूक्लियर डिवीजन होने लगता है, उसके बाद अल्टीमेटली बनते हैं मीरोजॉइड्स. क्या बनते हैं? मीरोजॉइड्स, ओके? स्पोरोजॉइड से हम इन्फेक्ट हुए, हमारे ब्लड के थ्रू वो धीरे धीरे लिवर में गए, लिवर में कुछ दिन आराम से रह के इट अलाउड द मल्टीप्लिकेशन साइकिल टू अकर. फ्रॉम देयर इट अलाउज द डेवलपमेंट ऑफ मीरोजॉइड. सो दिस इज हैपनिंग ऑल एसेक्सुअली. प्लीज रिमेंबर, दैट इज व्हाई आई टोल्ड यू इनिशियली इटसेल्फ, मैंने बोला था ना कि यहाँ पर एसेक्सुअल स्टेज होता है ह्यूमंस में, है ना? सो दिस इज एग्जैक्टली व्हाट आई एम एक्सप्लेनिंग राइट नाउ. सो कमिंग बैक टू दिस पॉइंट, अब ये जो मीरोजॉइड होते हैं, ओके? अच्छा, स्पोरोजॉइड और मीरोजॉइड में एक बहुत कॉन्सेप्चुअल पॉइंट ऑफ डिफरेंस ये होता है कि स्पोरोजॉइड जो होते हैं वो मोटाइल होते हैं एंड जो मीरोजॉइड्स होते हैं वो अमीबॉइड मूवमेंट दिखाते हैं थोड़ा सा, ओके? सो कमिंग बैक टू दिस पॉइंट, ये जो मीरोजॉइड बने हैं, अब ये जो मीरोजॉइट है अब ये थोड़ा सा डेवलप होना स्टार्ट कर जाएंगे एंड द मोस्ट इंपॉर्टेंट पॉइंट हियर इज दे आर नाउ गोइंग टू इन्वेड द ब्लड स्ट्रीम अगेन and cause infection there. Okay, what is going to happen here? These merozoites are going to leave the liver and enter the bloodstream by and causing infection in the red blood cell. एक बार जब वो red blood cell में आ जाते हैं तो हम उसको बुलाते हैं blood stage. यहाँ पर क्या होता है, ये जो merozoite है once again it will spend a few days, it will start multiplying again. merozoite से बनता है एक ring stage. अच्छा, इसको हम ring stage या फिर इनफैक्ट कई बार आप देखेंगे कि signet ring stage लिखा होता है. ओके, सिग्नेट रिंग स्टेज को क्यों बोलते हैं? क्योंकि अगर इस स्टेज पे आप लिटरली एक रेड ब्लड सेल को उठा करके माइक्रोस्कोप के अंदर देखेंगे तो आपको दिखेगा कि उसके अंदर एक रिंग बनी हुई है, लाइक लिटरली द रिंग दैट वी वेयर. ओके, देयर इज अ स्मॉल, लेट मी काइंड ऑफ मेक अ zoomed इमेज ऑफ इट हियर. तो एक सेल के अंदर, रेड ब्लड सेल के अंदर इस तरीके से रिंग स्टेज दिखता है. ठीक है, तो दिस स्टेज इज बेसिकली गोइंग टू डेवलप इनटू समथिंग कॉल्ड एज ट्रोफोजॉइड स्टेज. ओके, वंस अगेन स्पोरोजॉइट हमें इन्फेक्ट किया, लिवर में गया स्पोरोजॉइट, वहां पे उसका एसेक्सुअल साइकिल हुई, वहां से बने मीरोजॉइट वापस से ब्लड में इन्वेड कर रहे हैं. ब्लड में इन्वेड करने के बाद वो डेवलप होना स्टार्ट हो जा रहे हैं एंड बना रहे हैं रिंग स्टेज से होकर के अपना ट्रोफोजॉइट. एक बार ट्रोफोजॉइट बन जाएगा उसके बाद उसका और डेवलपमेंट होगा एंड फिर बनेगा एक फुल भरा हुआ समथिंग कॉल्ड एज शिजोंट. दिस इज अ वेरी 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 इंपॉर्टेंट स्ट्रक्चर. शिजोंट क्या होता है? ये जो ब्लड सेल है, वन ऑफ द ब्लड सेल, लेट्स से ये ब्लड वेसल है, यहाँ पर एक रेड ब्लड सेल है. इस रेड ब्लड सेल में बहुत सारे मीरोजॉइड्स डेवलप हो जाते हैं, बहुत सारे मीरोजॉइड. एंड ये जो ब्लड सेल है यूजुअली हमारी ब्लड वेसल पे अटैच हो जाती है, जहां पर भी ये माइग्रेट हो रहे हैं हमारे बॉडी में. Now this schizont is so full that as soon as this whole multiplication cycle is completed, this bursts open and releasing almost sometimes up to 32 merozoites also. Like one cell releasing almost 32 merozoites. Can you imagine that number? और ये जो 32 merozoites निकले हैं, अब ये और जाकर के बाकी सभी red blood cells को infect करेंगे. Do you know malaria is highly characterized by what? Fever, chills, shaking and sweating, right? This is the time, ट्रूली स्पीकिंग, जब शिजोंट बर्स्ट होते हैं और हमारे ब्लड में हर एक सेल को इन्वेड करते हैं. This is the stage when we actually get that spike of fever in malaria. You know that in malaria sometimes what happens is that हमें peak of fever आता है, फिर वापस से sweating होकर के थोड़ी से टाइम बाद कम हो जाता है, फिर peak of fever आता है, बहुत बढ़ता है fever, फिर कम हो जाता है. So why is this happening? This is happening because simply when the schizont is bursting, our body is getting infected, our further red blood cells are getting infected, and this is when our body, mounts an immune response to combat that, and that is why our body also raises temperature, and that is exactly what we see outside also, right? So ये हो गया हमारा वो शिजोंट स्टेज. ऐसे बहुत सारे मल्टीप्लिकेशन साइकिल्स होती हैं, बहुत सारे शिजोंट बर्स्ट होते हैं और फिर फर्दर मीरोजॉइड्स बनते हैं. अब जब ये ब्लड स्टेज चल रहा होता है, कुछ साइकिल्स ऑफ रेप्लिकेशन के बाद सम ऑफ द मीरोजॉइड गो एंड स्टार्ट एंटरिंग इनटू द सेक्सुअल साइकिल व्हिच इज बाय डेवलपमेंट ऑफ गैमीटोसाइट्स. देयर इज गोइंग टू बी मेल गैमीटोसाइट एंड फीमेल गैमीटोसाइट. अब इस टाइम पे जब भी कोई अनइन्फेक्टेड फीमेल एनोफिलस मस्कीटो, आई एम रिपीटिंग दिस पॉइंट, प्लीज ध्यान से सुनिए, इस स्टेज पे when few merozoids have developed into gametocytes, that is male and the female, this is female and this is your male gametocyte. अब यहाँ पर क्या होगा, whenever at this stage if a female anopheles mosquito which is not carrying plasmodium already visits us and sucks up the blood, in this blood this mosquito, uninfected mosquito, has taken up these gametocytes. These gametocytes will now travel into the gut, into the digestive tract of the mosquito. Okay, तो इस मस्कीटो ने, जैसा आपको दिख रहा है ये रेड कलर का ब्लड है इसके अंदर, इसने क्या किया, हमारा ब्लड सक कर लिया है, जिसमें हमारे पास तो गैमीटोसाइट्स थे अभी. नाउ दीज गैमीटोसाइट्स विल रीच अप टू द गट ऑफ दिस मस्कीटो एंड देयर इट विल डेवलप इनटू द मैच्योर गैमीट्स व्हिच विल अंडरगो फर्टिलाइजेशन. दैट इज व्हाई वी आर कॉलिंग इट एज सेक्सुअल साइकिल. एंड इससे बन जाएगा हमारा जाइगोट. जाइगोट विल फर्दर डेवलप एंड गेट कन्वर्टेड इनटू समथिंग कॉल्ड एज oocyst. It's not oocyte, it's oocyst. Please remember this point. And एक बार oocyst develop हो जाता है तो इसमें बहुत सारे sporozoids भरे हुए होते हैं. And these oocysts are going to then rupture, releasing so many sporozoids which are going to travel up till the salivary glands of the mosquito, right? Of the female Anopheles mosquito. And now again, see, this is where I started the cycle and I told you that I'll come back to this point. And this is when these sporozoids are going to be retransferred into some other human when this infected sporozoid carrying female Anopheles mosquito is going to visit a new human, uninfected human, right? And this is how the whole life cycle of the Plasmodium is going to get completed in the two hosts, primary being mosquito, the intermediate being the humans. Okay, तो इस क्वेश्चन के लिए आपका सही आंसर क्या होगा? The infectious stage of Plasmodium that enters the human body is, the answer to this is going to be sporozoids. Trophozoids कहाँ थे? Trophozoids blood stage में थे, वहाँ पर develop हो रहे थे. Then female gametocyte and male gametocyte कहाँ पे थे? ये भी blood में बन रहे थे, but these gametocytes were then picked up by the mosquito. Right? So I hope with that you all have understood this question and the whole life cycle of malarial parasite as well. Let us have a look at this question. Which of the following statements is correct? So you have been given 4 statements here and you have to choose which one is the correct statement. So one by one let us see. You can already see that the question is based on basically nitrogenous bases. If you try to revise it, nitrogenous bases are usually of 2 categories. There is purines and there is pyrimidines. Purines जो होते हैं ये structures होते हैं जिनमें 2 rings होते हैं, right? And pyrimidines are the structures which basically has 1 ring as a part of the structure, right? So 1 ring is there. All right. So now here when you talk about, especially in case of DNA, when you think of purines जो होते हैं वो होते हैं adenine and guanine, whereas pyrimidine are going to be cytosine and thymine. All right. अब यहाँ पर होता क्या है कि according to the Chargaff's rule, right, according to the base pairing principle, adenine, or rather I should say actually purine always pairs with a pyrimidine. Okay, and अगर वो प्यूरिन एडेनिन हुआ जो कि थायमीन के साथ हमेशा पेयर करता है तो ये जो पेयरिंग होती है दिस इज डन with the help of hydrogen bonds. And how many hydrogen bonds? This is the very specific principle. Adenine always pairs with thymine with the help of 2 hydrogen bonds. And जब भी आप देखेंगे cytosine, guanine and cytosine जो होते हैं वो पेयर करते हैं. Guanine is once again a purine and cytosine is a pyrimidine. तो ये जो पेयरिंग होती है ये पेयरिंग होती है with the help of 3 hydrogen bonds. Okay, इसीलिए शायद आपने जेनेटिक्स के चैप्टर्स में मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस में हमेशा पढ़ा है कि जब भी आप किसी ऐसे डीएनए मॉलिक्यूल को डीनेचर करने को ट्राई कर रहे हैं जिसमें जीसी कंटेंट ज्यादा है उसमें द अमाउंट ऑफ ईदर टाइम टेकन और मे बी द टेम्परेचर टेकन इज गोइंग टू बी हायर क्योंकि वहां पे जी और सी के बॉन्ड्स जो हैं वो ज्यादा होंगे, राइट? बिकॉज हाइड्रोजन बॉन्ड जो होता है वो एक टेम्परेचर सेंसिटिव बॉन्ड है, इसके लिए आपको टेम्परेचर प्रोवाइड करना पड़ेगा. अगर किसी डीएनए मॉलिक्यूल में जीसी ज्यादा हुआ तो ऑब्वियस सी बात है कि उस डीएनए मॉलिक्यूल को तोड़ने के लिए या सेपरेट करने के लिए, तोड़ने का मतलब है यहाँ पे डीनेचरेशन ऑफ द 2 strands, यहाँ पर यू विल हैव टू गिव इन मोर एनर्जी इन कंपैरिजन टू अ डीएनए मॉलिक्यूल व्हिच हैज मोर नंबर ऑफ 80 bonds, right? Adenine and thymine bonds. Okay, so coming to the statements of this particular question, adenine pairs with thymine through 3 hydrogen bonds. Nahi. Adenine does not pair with thymine. No, this is an incorrect statement. Adenine pairs with thymine through 2 hydrogen bonds. Yes, this is a very correct statement. And adenine pairs with thymine through 1 hydrogen bond. Nahi. 1 hydrogen bond wali pairing to kisi ke bhi beech mein nahi hoti hai. So this is an image which basically shows you your thymine bond here. Let me change the color. This is where is your thymine, right? And this is your adenine molecule, right? ओके, सो यहाँ पर चाहे यूरासिल भी हमने लिस्ट किया, क्यों? क्योंकि कभी कभी क्या होता है आरएनए में फोल्डिंग होती है एंड यू नो दैट इन केस ऑफ आरएनए थायमीन इज रिप्लेस्ड विद यूरासिल. सो दिस पर्टिकुलर pyrimidine is going to pair with adenine with the help of 2 hydrogen bond. This dotted line that you see, you know hydrogen bonds are usually always shown with partial line, है ना? तो ये हाइड्रोजन बॉन्ड एक हुआ और ये हुआ आपका दूसरा हाइड्रोजन बॉन्ड. सो इस क्वेश्चन के लिए व्हाट इज गोइंग टू बी द राइट आंसर? ऑप्शन सी. आई होप यू ऑल गॉट दैट क्वेश्चन करेक्ट. Let us have a look at this question. Flippers of penguins and dolphins are examples of. Okay, so now when you think of penguins and dolphins, here the flippers are developing from because these two organisms are not really related, hence their development of these flippers are also completely different, right? So development of these kind of things, when it is happening independently in kind of unrelated organisms, what is it called as? It's called as the analogous organs, right? You remember this part, you've learned about this in evolution chapter, right? So analogous organs, development of them basically represent convergent evolution, right? Why? Because simply when you see distinct or not even related organisms develop structure which have similar functioning, what is happening? They are basically coming to one common point. They are basically converging at one common point, and this is why we represent this kind of evolution method as convergent evolution. Okay, and when we see organs which are having similar origin but their functions are different, then we call those organs as homologous organs, and those ones represent divergent evolution. Okay, so coming to this question, obviously the right answer is going to be option D, but let me quickly remind you what is industrial melanism, what is natural selection, and what is adaptive radiation, because of which these are not the right answer. Starting with the industrial melanism first. Ye hota kya hai? This is basically an evolutionary effect only, where you see that there is development of certain organisms with dark pigmentation, etc. And this because it happened in response to industrialization, especially when it was seen initially in England. So this is when this whole phenomena was also studied. This whole thing is referred to as industrial melanism. Okay, so let me show you the image of these moths which actually developed in response to and adapted better in response to this whole industrial pollution. When we talk about industrial pollution, we are usually referring to gases like sulfur dioxide, which is evolved. We are also referring to dark soot that is released because of lot of release of carbon, right? So at that point of time, what used to happen was that there was a wild variety of moth, like you see the grayish colored one. This is called as Biston betullaria, right? Now this is the wild variety of the moths that was seen at that point of time. Before that point of time, because these particular moth had a beautiful adaptation by camouflaging themselves with respect to the color of the bark of the tree. Now when the industrialization started happening, when this whole revolution was happening, there was a lot of release of these toxic gases, these poisonous and polluting gases that were coming out. They were forming darker colored tree trunks because of simply deposition of all these carbon soot. Okay, so now what happened is that at this time, what was happening was that Biston betullaria became very, very visible over the darker background, and hence they were easily available for predation. Okay, they were easily eaten up by their predators. At the same time, what happened was that these dark colored ones that you see here, okay, this is what is called as, let me write it down here, this is what is called as Biston carbonaria, obviously because of the dark color and carbon which was coming. The soot comes because of carbon here. So these ones, these dark colored moths basically had better survival at that time, and that led to the increase in the count of Biston carbonaria in comparison to Biston betullaria. So this whole story is referring to industrial melanism. The next option that you had, and obviously that is why that option was wrong, the next option that you had was basically on natural selection. So you know that natural selection, what have you learned? Natural selection basically refers to a property where due to evolutionary changes, only the organisms which survive better are going to be selected by the nature, right? So now one of these examples that you've learnt about is, the theory of Lamarck was given, then there was a theory of Darwin that was proposed. Now here what you're seeing is that long neck of the giraffe is said to be a result of natural selection. Okay, now what is happening here is that the long neck giraffe had better access to food, and apparently that is why they had better survival and better reproductive capacity, and once again because of that they had better survival. So this whole point is natural selection. They are not related to flippers of penguins and dolphins, etc. So that makes this option incorrect. Talking about this beautiful example that you have learned once again in evolution chapter, which is called as adaptive radiation. Now what happens in adaptive radiation is that this is once again an evolutionary process जहां पर आपने पढ़ा है कि Darwin's finches के बारे में, right? So Darwin's finches, which were seen on Galapagos Island, they basically came at some point from a common ancestor from South America, migrated to this Galapagos Island, and because of them occupying different kind of habits, different kind of niche in that particular island, what happened was that they developed over a period of time with different feeding habits. Some of the finches were feeding on, they were basically pecking wood, so the wood pecking finch would have a different kind of beak. Then the ones that were feeding on insects, like warblers are there, then they were the ones who were feeding on the seeds, etc. So what my point here is that due to these different feeding habits, these particular finches developed from one common ancestor into these different structures of beak having finches, right? So this kind of evolution represents what is called as adaptive radiation, and that is why this will not be the right answer. Going ahead further, you see here that we have shown you an image of a parrot which belongs to class Aves. Then we have bats, which is a mammal. Then we have butterfly, which belongs to altogether non-chordates, right? We are talking about basically the phylum Arthropoda. So butterfly, insects there, right? So all these organisms, completely unrelated ones that you're seeing here, they have one common adaptation, which is of flying, right? And this flying is basically happening from different parts of their body, and hence the different origin of their wings, right? So this is exactly what represents the convergent evolution, right? This is another example of the question that we have been asked. And talking about penguin, which belongs to class Aves, which are birds, and dolphins, which are mammals, they also have flippers, which are useful in swimming, and hence they are representing the convergent evolution. So what is going to be the right answer for this question? It's going to be option D, that is convergent evolution. Let us have a look at this question. Montreal Protocol was signed in 1987 for control of. अब ये बहुत ज़्यादा एक important protocol है जो कि sign हुआ था in order to control and limit the emission of ozone depleting substances. ठीक है, ये जो है this was signed in 1987 and this is a very important protocol when you talk about your environment. And I'll tell you the one point where maximum children got confused here, and because of the lack of once again conscious efforts in understanding the difference between the right concepts, they marked option A as the right answer. Now you have to remember there is another important protocol that is there, which is called as Kyoto Protocol. Right? ये जो है ये 97 में आया था, 1997. Let me write it down this way. Yeah, this was given in 1997 and this was done in order to control the emission of greenhouse gases. ठीक है, तो आप ये point of difference जो है बहुत अच्छे से याद रखिए. क्वेश्चन में मॉन्ट्रियल प्रोटोकॉल के बारे में पूछा गया था. This was once again, I am repeating this was an international treaty which was signed by so many nations and so many countries in order to control the emission of ozone depleting substances. And ये हुआ क्यों था? क्योंकि actually 1985 में finally scientists वगैरह ने ये देख लिया था कि that there was a huge gap that was being created in the ozone layer, especially over the Antarctic region, right? So because of this in 1987, with a lot of hush-hush, this whole protocol came out in order to start controlling the amount of ozone depleting substances. Kyoto Protocol came much later, and that was in 97, which was responsible for controlling the release of greenhouse gases. Let us quickly discuss all these options a little bit more in detail. So पहली चीज, जैसा मैंने आपको बताया, Montreal Protocol, it was an international treaty which was designed to protect the ozone layer, especially because there was an ozone hole that was seen over the Antarctic region, right? And this is very important क्योंकि आपको पता है ओजोन जो है बहुत सारे harmful radiations को absorb करती है. This is done in order to kind of protect the whole ozone layer in the stratosphere. Okay, stratosphere. Now going ahead, emission of greenhouse gases, मैंने आपको already answer बता दिया. इसके लिए जो protocol है वो होता है हमारा Kyoto Protocol, which was given in 97. इसको भी आप याद रख सकते हैं. फिर आया था हमारा option which was for transport and handling of genetically modified organism. तो इसके लिए एक different protocol है जो आपके syllabus में नहीं है, but let me just state out its name, which is called as Cartagena Protocol. अब ये जो है, this protocol basically takes care of all the legal issues which deals with handling of the genetically modified modified organism, transport of it, and if there are any ethical or safety issues which are associated with the GMOs. Okay. Next is your e-waste was given. What is e-waste? E-waste होता है जब कोई भी electronic device after a certain time when it is not at all in use, you cannot refurbish any of its remaining part, right? So this whole waste is called as e-waste, and इसके management के लिए usually every nation has a different set of guidelines, which is what is called as e-waste management guidelines as such. So every different nation has a different protocol, and Montreal protocol does not have anything to do with e-waste management. So the right answer for this question is going to be option D. चलिए इस क्वेश्चन को देखते हैं. आइडेंटिफाई द रॉन्ग स्टेटमेंट विद रिगार्ड टू रेस्ट्रिक्शन एंजाइम. तो रेस्ट्रिक्शन एंजाइम्स के बारे में कुछ स्टेटमेंट्स दिए गए हैं एंड वी हैव टू चूज द रॉन्ग स्टेटमेंट हियर. पहला स्टेटमेंट बोलता है दे आर यूजफुल इन जेनेटिक इंजीनियरिंग. यस, वेरी मच. यही तो हमारे मॉलिक्यूलर सीजर्स हैं, एंड इफ मॉलिक्यूलर सीजर्स वर नॉट देयर वी कुड नॉट कट द DNA, right? We could not cut the gene and do whatever required cloning has to be done. So obviously this statement is a very, very correct statement. It's not the incorrect statement. Next statement है sticky ends can be joined by using DNA ligases. तो एक्चुअली इस स्टेटमेंट की ऑथेंटिसिटी सिर्फ हम आइसोलेटेड स्टेटमेंट की तरह देखेंगे तो ये ऑब्वियसली सही है. क्यों? क्योंकि स्टिकी एंड्स जो होते हैं, जो डेवलप होते हैं व्हाइल कटिंग द DNA, दे आर डेफिनेटली जॉइंड बाय DNA ligases. बट आर दे अ प्रॉपर्टी ऑफ रेस्ट्रिक्शन एंजाइम्स? नो, राइट? तो इसलिए ये जो स्टेटमेंट है, एंड दिस इज एग्जैक्टली व्हाट द कंफ्यूजन दैट मैक्सिमम चिल्ड्रन हैड, है ना? तो ये जो स्टेटमेंट है ये गलत स्टेटमेंट है विद रिस्पेक्ट टू रेस्ट्रिक्शन एंजाइम. आइसोलेटेड स्टेटमेंट की तरह ये सही स्टेटमेंट है, पर क्वेश्चन ने क्या पूछा था? हमें विद रिगार्ड टू रेस्ट्रिक्शन एंजाइम्स देखना, and that is why option B is going to be the correct answer here, right? It's an incorrect statement with respect to restriction enzymes. आगे के स्टेटमेंट पे चलते हैं. Each restriction enzyme functions by inspecting the length of DNA sequence. Yes, very much. Restriction enzyme क्या करते हैं? पूरे DNA sequence को स्कैन करते हैं, फिर उसमें अपना recognition sequence ढूंढते हैं जो यूजुअली एक पैलेंड्रोम होता है, और वहां पर restriction साइट पे कट करते हैं. यही नॉर्मल फंक्शनिंग होती है, राइट? इसके लिए मैं आपको एक बहुत सुंदर सी इसके बाद फ्लो शीट इमेज फॉर्म में दिखाऊंगी ताकि आपको समझ में आए कि रेस्ट्रिक्शन एंजाइम की फंक्शनिंग कैसे होती है. तो ऑब्वियसली ये एज अ स्टेटमेंट करेक्ट है विद रिस्पेक्ट टू रेस्ट्रिक्शन एंजाइम. एंड चौथा स्टेटमेंट जो यहाँ पे दिया गया है, दे कट द स्ट्रैंड ऑफ DNA at palindromic sites. Yes, what are palindromes? They are basically the sequences which can be read in a similar fashion when you read it from both the sides. Like one of the best examples to understand language palindrome sequences, Malayalam, right? Malayalam as a language, when you spell it out, it's read the same way when you read from left to right or from right to left. Okay, now let us quickly understand these statements with respect to the whole functioning of restriction enzyme. So this is like a small flow sheet where you see how genetic engineering is done using restriction enzyme. What you do is, let me change the pen color first. Yes, what you do here is that you take the DNA sequence, required DNA sequence after PCR, after making multiple copies of it, it's going to be digested or cut with the help of restriction enzyme, and it is going to be cloned inside the vector which is cut by the same enzyme. You all know this principle. This is very well taught to you in the biotechnology applications chapter, right? Principles, of course, you've used the whole principle there, and then how to apply it you've learned in application chapter. Now once this ligation happens, this whole recombinant DNA is going to be used for transforming a bacteria from where we are going to upgrade the product and upscale the product that we are using. Okay, going ahead. So here is a palindrome sequence which you can see very clearly. Now here the sequence that we've given is GAA TTC, है ना? This is 5 prime, this is 3 prime. When you see the copy of it on the complementary strand, you would see this is read in the same way, GAA TTC, right? That is why we called it as a palindrome. Okay, now this is a recognition sequence which is read by an enzyme which is called as EcoR1. Okay, EcoR1 is the name of the restriction enzyme which we obtain from E. coli bacteria. Restriction enzyme 1. Okay, so now the reason why I have listed this example so that you can understand how restriction enzyme exactly work. So what you see here is a palindrome sequence which lies in the recognition sequence, and here at specific site, that is between G and A, is where the restriction enzyme is actually going to cut. Which restriction enzyme? This specific restriction enzyme, EcoR1 enzyme. Okay, now what happens next is when this restriction enzyme binds to that particular sequence, it is going to cut at the respective position which I told you, right? generating overhangs like this. Okay, अब यहाँ पर होता क्या है कि आपको बहुत क्लियरली दिख रहा है जब भी कोई रेस्ट्रिक्शन एंजाइम कट करता है, आपने पढ़ा है कि यूजुअली या तो ब्लंट एंड जनरेट हो सकते हैं या फिर स्टिकी एंड्स जनरेट हो सकते हैं. स्टिकी एंड को हम स्टिकी क्यों बुला रहे हैं, यहाँ पर आपको दिख रहा है कि इस एंजाइम ने जब कट किया, बिकॉज ऑफ देयर कटिंग साइड्स, बिकॉज ऑफ इट्स रेस्ट्रिक्शन साइड, व्हाट यू हैव सीन हियर इज दैट यहाँ पर कुछ इस तरीके से ओवरहैंग्स बच गए हैं, है ना, कुछ एक्स्ट्रा पोर्शन है जो सिंगल स्ट्रैंडेड है. इसकी वजह से, बिकॉज यू नो दैट दीज बेसिकली ओवरहैंग्स हैव कॉम्प्लीमेंट्री बेस पेयर, right? This A will pair with T, this T will pair with this A will pair with this T, and this T can pair with this A. तो अब क्या हो रहा है, इसकी वजह से क्या होता है कि बहुत easily इसका ligation हो सकता है, right? With the help of DNA ligase enzyme, of course. Okay, now that is why we are calling this as sticky ends. दूसरा ऑप्शन क्या होता है, कई बार क्या होता है कि DNA जो strands हैं वो इस तरीके से कटते हैं, which basically does not leave any overhangs like this, right? And that is why we call them as blunt ends. These are called as blunt ends. वहाँ पर कोई भी overhangs नहीं होते हैं. So coming back to the sticky ends, yes, restriction enzymes most of the times, especially the ones that are used in genetic engineering, generate sticky ends. If they don't, let us say if they produce blunt ends, we actually add overhangs to them in order to allow the further ligation to happen. Okay, तो अब यहाँ पर क्या होगा, यहाँ पर ये जो sticky ends है इनको हम लोग DNA ligase नाम के दूसरे एंजाइम से चिपकाने वाले हैं, ligate करने वाले हैं. And that is why I told you all कि ligation DNA ligase जरूर करता है, but that's not a property of restriction enzyme, which was what was question asking about, right? So here you have ligation of the sticky ends with the help of enzyme DNA ligase. So what is going to be the right answer for this question? Wrong statement पूछा गया था. It's going to be option B. Let us have a look at this question. By which method was a new breed Hizardale of sheep? So this is a new breed of sheep, right, which is formed by using Bikaneri ewes and Merino rams. Okay, तो पहली चीज, in this question, like many children get confused between what exactly is an ewe, what exactly is a ram. They think that they are altogether different species. This is very simple. Rams basically refers to the male sheep, okay, and ewes is nothing but sheep only, but it is the female sheep. So first thing is, please remember these terms, okay. So female sheep are called as ewes and male sheep are called as rams. Now here, these two varieties that you are seeing, Bikaneri and the Merino ones, these are basically different breeds, and that is why the answer for this question is going to be crossbreeding. But this is one question where children find it very confusing to understand what is inbreeding, what is outbreeding, what is outcrossing, what is crossbreeding, what is interspecific hybridization, what is mutational breeding, etc. Okay, so let me just put an end to that particular confusion and let me explain you this thing very beautifully in the form of a flow sheet so that you all can retain this information very well. This is an important point when you talk about animal husbandry, etc., which basically belongs to your chapter strategies of enhancement in food production, right? So let me remind you this particular flow sheet, which is very well given in the textbook also, but let us quickly try to revise it all together. Okay, so when you talk about breeding, what is breeding? When you allow reproduction to happen between two different organisms, right? When I say different organisms, I am not referring to a species or a different genus or things like that. I am just talking plain simple English where reproduction is going to happen between two individuals. Okay, now this breeding can be of two types. There is going to be inbreeding, there is going to be outbreeding. Okay, what is inbreeding? Whenever you allow the breeding to happen between related individuals. Okay, what are related individuals? Which basically means we have checked their history for about 4 to 6 generations at least. Okay, so mating of closely related individuals within the same breed. You will remember this point very well. It is always going to be within the same breed when you talk about inbreeding. Okay, and how closely related individual? We are looking at 4 to 6 generations. Okay, clear everyone? So this is called as inbreeding. Now normally what happens is that this kind of thing allows the indigenous quality to be maintained, but what happens after some time is that due to continuous inbreeding, because of lack of any contribution with respect to evolution as such, this kind of leads to loss of vigor, loss of lesser amount of fertility, lesser amount of productivity in future generation. So in order to overcome this whole thing, which is called as inbreeding depression, outbreeding is done. That is this thing, right? This term that I have given here, outbreeding is done. What is outbreeding? Outbreeding is when you are going to allow the mating to happen between unrelated individuals. Now they can be of the same breed, they can be of different breed, they can be of different population, they can be of different species also, right? So breeding of unrelated animals is what is called as outbreeding. Okay, why is it done? It can be obviously done in order to attain a specific type of variety if you want, or it can be done in order to overcome the inbreeding depression. Now this outbreeding is of further, majorly speaking, three types. First is outcrossing. It's like the simplest form of outbreeding. यहाँ पर क्या होता है? Mating is done between unrelated members but of the same breed. Very important point. You are using the same breed, but it has to be unrelated individual for at least 4 to 6 generation. Here also the same principle applies. Okay, for at least 4 to 6 generation. So you remember inbreeding में हमने क्या किया था? 4 to 6 generation तक उनके individuals related होने चाहिए थे. यहाँ पर हम क्या कर रहे हैं? Breed same ले रहे हैं, but we are using unrelated individuals by checking their history of up to 4 to 6 generation. Next thing is crossbreeding. Actually, this is the most specific technique which is used to overcome inbreeding depression. In fact, many times you get a question that which of the following outbreeding methods is used to overcome the inbreeding depression. अगर आपको इन सब में से choose करना हो तो आप ये आंसर crossbreeding का choose करेंगे. Okay, crossbreeding क्या होती है? It is basically mating of superior variety or superior males of one breed and सुपीरियर फीमेल ऑफ द अदर ब्रीड. इसकी वजह से जो वैरायटी होगी उसमें प्रोडक्टिविटी काफी ज्यादा हाई होती है. सो मेटिंग ऑफ सुपीरियर मेल्स ऑफ वन ब्रीड विद सुपीरियर फीमेल्स ऑफ अनदर ब्रीड. तो हिजारडेल जो है वो इसी क्रॉस ब्रीडिंग से आई है. ओके, क्रॉस ब्रीडिंग से आई है, आया है, डिपेंडिंग अपॉन यू नो व्हाट सेक्स इज गोइंग टू बी हैपनिंग फॉर्मिंग इन द फ्यूचर जनरेशन. देन कम्स योर इंटरस्पेसिफिक हाइब्रिडाइजेशन. इंटरस्पेसिफिक हाइब्रिडाइजेशन क्या होता है? व्हेन देयर इज हाइब्रिडाइजेशन, दिस इज गोइंग टू बी एन आर्टिफिशियल प्रोसेस व्हिच इज डन बिटवीन डिफरेंट स्पीशीज. व्हेन वी से इंटरस्पेसिफिक, इट मींस यू आर गोइंग टू यूज 2 डिफरेंट स्पीशीज. एंड स्पीशीज को बाय डेफिनेशन आप जानते हैं, इट इज दैट ग्रुप ऑफ ऑर्गेनिजम्स व्हिच कैन इंटरब्रीड. अगर आप 2 स्पीशीज ले रहे हैं तो वो नेचुरली तो इंटरब्रीड नहीं कर पाएंगे. सो ऑब्वियस सी बात है, यहां पर 2 डिफरेंट स्पीशीज की हम आर्टिफिशियल इंटरब्रीडिंग कराते हैं. Okay, clear everyone? Mule is one example of interspecific hybridization. Okay, then comes your answer here. Merino ram लिया हमने, which is the male, right? Let me write it down here for you. फिर हमने ली बीकानेरी ईव, which is a female sheep, and then we allowed their mating to happen, leading to formation of Hizzardale. Okay, समझ में आया सब लोग को? This is what is called as cross breeding. And here there is one option which actually most of the children miss once again. Mutation breeding में क्या होता है? It's once again an artificial technique जहाँ पर आप using chemical mutagenic agent etc. you allow some changes, some mutations, some desired mutations to happen. Usually हम mutation breeding plants में, seeds में यहाँ पर कराते हैं. So we allow that mutation to happen and that is what is bred in further generation. That is what is called as mutational breeding. So obviously इस क्वेश्चन के लिए ये वैलिड नहीं है. The right answer for this question is going to be option A, that is cross breeding. Let us have a look at this question. Which of the following refer to correct examples of organisms which have evolved due to changes in environment brought about by anthropogenic action? Okay, what is the meaning of anthropogenic action? It basically refers to the actions that are man-made, human-made, right? जो सब contributions हमारी वजह से हैं in the environment because of which evolution or changes have happened here and it has led to development of new organisms or new adaptations. That is what this question is asking all about. Okay, तो यहाँ जो options दिए गए हैं वो है पहला example, that is Darwin's finches of Galapagos. ये तो आप लोग जानते हैं कि Darwin's finches is a beautiful example of natural selection and not of anthropogenic action, right? क्यों? क्योंकि Darwin's finches जो हैं, ये जो birds थी, finches थी, ये basically एक दूसरे continent से आई थी on the Galapagos Island and when they came here due to them occupying different habitats in that particular location, like कुछ ने tree के different spaces को occupy किया, कुछ लोगों ने, कुछ finches ने land को occupy किया, तो ये सब होते होते क्या हुआ कि उनकी जो beaks हैं, they basically adapted, they basically developed into newer and different structures in order to allow this kind of feeding habit to continue and in order for them also to survive a little better. Okay, तो ये जो है ये right answer नहीं हो सकता है. दूसरा ऑप्शन दिया गया है herbicide resistant weeds. तो yes, आपको तो पता ही है कि DDT का स्प्रे recent times में इतना ज़्यादा बढ़ गया था that because of that there is a development of lot of DDT resistant mosquitoes etc. and flies etc. because of which the predominance of malaria also increased, right? I'm just giving you one overview of example, but there are so many examples when you look at in the environment that because of these continuous use of herbicides, different types of herbicides, in fact pesticides and insecticides also, when we use on different weeds, what happens is herbicide we use for weeds, but pesticides we use for pests as such, and insecticides for insects. So when we use these chemicals over a repeated period of time in large quantities, it leads to development of resistance, right? So this is, yes, completely man-made because this is what we have produced and we have been using in our environment. So this is a correct statement with respect to the question. Then comes drug-resistant eukaryotes. So drug-resistant eukaryotes, when we talk about here, actually drug resistance majorly you can understand in prokaryotes first so that you understand its effect on eukaryotes. Think of example of tuberculosis, right? Now tuberculosis in a country like India, it is pretty much a very, very prevalent kind of a disease, right? It's an infectious disease. But we did find a cure to it by, getting medicine, etc. There is rifampicin, etc., which is used for treatment of tuberculosis. But then what happens here is that because of overuse of medicines, what has happened is that lately you must have heard of so many cases where there is multi-drug resistance tuberculosis that is developing. In fact, even after, multi-drug resistance, there is this super resistant species that has evolved even after that. And that kind of thing is leading to development of drug resistance, especially in prokaryotes, like I'm talking about bacteria here. Similarly, when we talk about humans, like any eukaryotic cell for that matter, if we take any medicine, especially when we talk about antibiotics, etc., it affects our flora, our microorganisms, the bodily microorganisms so much. Once again, this is predominantly at prokaryotic level, but this kind of drug resistance do develop over a period of time when you have continuous exposure to certain medicine, right? So this is also completely man-made because we know that we are the people who have created all these medicines and consuming it. Of course their origin is natural, but over exploitation of that is what has led to drug resistance. Then we have another option here which says man created breeds of domesticated animals, like for example dogs. So we use them as pets as such. There are so many other examples of domesticated animals like sheep, etc. So yes, this is completely man-made. So if you look at the correct answer for all these options, it's going to be option A, which means B, C, D are correct. So let me very quickly show you the relevant, beautiful images that we've made for these things. Evolution by anthropogenic action is due to the interference of human beings. We have used dogs and produce so many different breeds of it. Now if you go to someone's house, you would see that this person has this breed of dog, and then that person has different breed of dog. They have different breeds of cats as such. When we talk about the pet animals, there is different breeds of cows that we have domesticated, breeds of sheep, etc., and whatnot, right? So this is what has happened due to anthropogenic actions. Then we have drug resistance in eukaryotes, like I already specified. We have herbicide resistant weeds that are coming up due to over exploitation. But Darwin's finches, like I was telling you, different structures of their beak, as you are seeing, is developing because of natural selection, because of their own adaptation to the changes in the environment. And what is the environmental change that has happened here is the change of the food that they were receiving over a period of time, because of which their beak also adapted, right? So the right answer here is going to be option A, that is B, C, D option. Let us have a look at this question now. Meiotic division of secondary oocyte is completed. Now this is a very straightforward question from the chapter Human Reproduction. Alright, so meiotic division of the secondary oocyte is completed after zygote formation, at the time of fusion of a sperm with an ovum, prior to ovulation or at the time of copulation. Okay, so here the answer is very clear. The answer is going to be option B. Because after zygote formation, no, this is when the fertilization has already happened and the zygote has been formed. So no, the second meiotic division of the secondary oocyte happens before this. Prior to ovulation, no. At the time of copulation, no. Now this is one confusing option that the children felt, but the point is that there is a very, very clear option than this option. So when the copulation is happening, obviously sperms are going to get released, but only when the sperm attaches to the ovum in order to allow fertilization to happen. So that start is what is going to lead to the meiotic division, completion of meiosis in the secondary oocyte. I'll very quickly show you this beautiful image which just shows you the whole flow sheet of this kind of development. So you all know, let me just change the color. Yeah, so you all know that before birth the oogonium develops or mitotically divides in order to form the primary oocyte, which before birth is going to get arrested at first meiotic stage. So the meiosis starts, but then it gets arrested there itself, right? Then when the puberty comes, this meiosis is going to continue and form the secondary oocyte. But this secondary oocyte also where second meiotic division starts, but it arrests. Okay, so this is a very important point, that in the secondary oocyte, the meiotic division, the second meiotic division also starts, but it stops or arrests at the metaphase 2 stage. Very important point. What is metaphase 2 representing? It is representing the metaphase of meiosis 2, which is the second part of meiosis, right? So when ovulation happens, egg is going to be released, and very important point, if the sperm is released, then after sperm entry, the meiosis is going to be triggered in this, the remaining meiosis is going to be triggered in this secondary oocyte. So what has happened? You have seen that when the egg is there, when sperm has attached to it just before fertilization, this event is going to happen, and once this happens, the meiosis is completed, the fertilization would occur, and it will lead to the formation of zygote, ultimately the fertilized egg, right? So the right answer for this question is going to be option B, that is at the time of fusion of sperm within ovum. Let us have a look at this question. In relation to gross primary productivity, which we also call as GPP, okay, and net primary productivity of an ecosystem, so we are talking about NPP here, okay, which of the following with respect to GPP and NPP is correct out of the given statements? पहली चीज, it's a very straightforward question. GPP होता क्या है? What is gross primary productivity? It is basically the rate at which for an ecosystem the carbon is getting fixed, right? So simply speaking, it is going to be the rate at which organic matter is synthesized by the plants, the total organic matter. ओके, लेकिन ऑब्वियसली बात है, प्लांट खुद एक लिविंग ऑर्गेनिज्म है, तो उसको जितना भी अमाउंट ऑफ कार्बन वो फिक्स कर रहे हैं प्लांट्स, उसको कुछ तो अमाउंट खुद के लिए यूटिलाइज करना होगा. इट विल रिक्वायर सम अमाउंट टू बी यूटिलाइज्ड इन द रेस्पिरेशन प्रोसेस. तो इन प्रोड्यूसर से, इन प्लांट से जितना अमाउंट ऑफ ऑर्गेनिक मैटर जो है अवेलेबल रहेगा बाकी ऑर्गेनिजम्स के लिए, दैट इज व्हाट इज कॉल्ड एज द नेट अमाउंट, है ना? क्यों? क्योंकि टोटल अमाउंट में से खुद प्लांट्स यूज करेंगे, जो बचेगा बाकी ऑर्गेनिजम्स के लिए, दैट बिकम्स द नेट अमाउंट, राइट? सो सिंपली स्पीकिंग, ग्रॉस इज गोइंग टू बी मच हायर देन द नेट प्राइमरी प्रोडक्टिविटी विच इज अवेलेबल फॉर अदर कंज्यूमर्स टू बी टेकन, राइट? नाउ लेट अस रीड द ऑप्शंस वन बाय वन. Gross primary productivity and net primary productivity are one and the same. No, I just told you the difference. There is no relationship between gross primary productivity and net primary productivity. Yes, there is very much a good relationship that exists, so this option is outrightly incorrect. We have gross primary productivity is always less than net primary productivity. No, it is always going to be higher, क्योंकि gross is what we are referring to, the total amount that is fixed by the plants in an ecosystem, right? तो ये statement भी गलत हुआ. ऑब्वियसली बात है, एक ही बचा ऑप्शन हमारे पास. ग्रॉस प्राइमरी प्रोडक्टिविटी इज ऑलवेज मोर देन द नेट प्राइमरी प्रोडक्टिविटी. यस, वेरी मच. लेट मी शो यू अ वेरी क्विक इक्वेशन फॉर दिस so that you understand better. So gross primary productivity is the total amount of carbon that is fixed by the plants, total amount of organic matter that is going to be synthesized by the plants. This is always going to be equal to the net amount, which is amount of carbon, fixed carbon, which is available to the consumers at the next level of the ecological pyramid, right? Plus whatever is the one that is going to be लॉस्ट ड्यूरिंग रेस्पिरेशन फॉर द प्लांट. सो प्लांट ने खुद ने जितना लूज किया है, जितना वो यूज करने वाले हैं दैट अमाउंट, प्लस जितना भी अवेलेबल है अगले इकोलॉजिकल लेवल के लिए, है ना. सो दिस होल थिंग विल सम अप टू ग्रॉस प्राइमरी प्रोडक्टिविटी. सो व्हाट इज गोइंग टू बी द राइट आंसर? इट्स गोइंग टू बी ऑप्शन डी फॉर दिस क्वेश्चन. So children, that was about today's discussion for NEET 2020 paper. I hope आप सबको सभी questions अच्छे से समझ में आए, सब question से related सभी concepts भी आपके अच्छे से revise हो गए. Try practicing this way itself, right? So I'll see you all next time. Till then, keep learning.